All right, so we're going to talk about the structure of DNA. So where we left off was talking about Franklin and her discovery of the picture, so showing the double helix structure, which you can see in the background on the slide, but also Watson and Crick, who actually developed that very first model. All right, so DNA, like I said, is referred to as a double helix. So when you think of the word double, you need to think two. And you think of helix, you need to think basically twisted. So it looks like a twisted ladder, a spiral staircase. All right, and the building blocks of that DNA molecule are referred to as nucleotides. So if you think back to module 2, we talked about nucleic acids, and we said that they are made of nucleotides. So a nucleotide is consisting of 3 different parts. One, a sugar called deoxyribose, 2, a phosphate group, and 3, one of the 4 nitrogen bases. So there are 4 possible nitrogen bases in a DNA molecule. Let me try to hide that down there. All right, so we've got adenine, which is represented by the letter A, thymine, which is represented by the letter T, guanine, which is represented by the letter G, and then cytosine, which is represented by the letter C. All right, so if we're looking at our DNA structure, and I kind of popped that picture down at the bottom of this slide. All right, so we've got our C's and our G's are going to pair together. So our cytosines and our guanines fit perfectly together like little puzzle pieces. Our adenines, our A's, and our thymines, our T's, hook together the same way as that puzzle piece. So whenever you see a C, it's going to pair up with a G in the DNA molecule. Whenever you see an A, it's going to pair up with a T. All right, so there are 2 strands of nucleotides, and I've put up this big giant picture no, let's point correctly at it. How do I point at this? This way, this way, this way, this way. All right, so this particular DNA molecule is showing you those A's and those T's paired together by hydrogen bonds. And these hydrogen bonds are so important because they are weak bonds. So these are very weak chemical bonds. And when you think of something being weak, you think of it as being easily broken. All right, and this is going to play a huge part when we start talking about DNA replication because these hydrogen bonds have to break at some point. All right, so these hydrogen bonds, these weak bonds, are holding these nitrogen bases together, those A's and those T's, those C's and those G's. All right, and these nitrogen bases bond in a very specific way. Like I said, your A's and your T's always go together. Your G's and your C's always go together in DNA. And this is referred to as complementary base pairing. All right, so if we're practicing this, when given one side of a DNA strand, you can actually determine what the other side looks like based on that rule of complementary base pairing. So if we've got a T, it's going to match up with an A. All right, so looking at this strand right here, all right, so we've got, for example, we've got a T, A, G, G, C, T, G, A, C. All right, so our very first T is going to match up with an A. So on your paper right now, I want you to copy down this base pair strand, pause the video right now so that it's not going past that point, and write down that TAGGCTGAAC. All right? Once you have done that, I want you to write down the complementary base pairs. All right? So T will always pair up with A will pair up with T, G will pair up with C, G will pair up with C. All right, so if we want to write the complementary base strand, you should have written down A, T, C, 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 G, A, C, T, T, G. All right, so this particular sequence above only has 10 base pairs, so only 10 sets of letters in there. If we're talking about a true human DNA strand, a human has approximately 3 billion base pairs in their DNA.